My name is Joseph Franses. I'm a medical oncology fellow at Dana-Farber and Mass General in Boston, and a postdoc in the lab of Dr. David Ting at the Mass General Cancer Center. And my talk today was describing our work studying the impact of this protein called Lin28B in pancreas cancer. So pancreas cancer is poised to become the second leading cause of cancer death in the United States within the next decade. There are multifactorial reasons for this, but suffice it to say that new insights into the biology of the disease will be critical to design the next generation of therapeutics and diagnostics. At Mass General, we have particular expertise in circulating tumor cell detection and characterization. And one of my first tasks in the lab was to characterize using RNA sequencing the CTCs, circulating tumor cells, released from pancreas cancer patients into the blood. And so from these cells, I characterized their sequencing profiles and found robust expression of this gene called Lin28B that I'm going to talk to you about a little bit more. So this gene is expressed mainly in developing embryos, but whenever it's present in human tumors, it correlates with poor outcome. And so I wanted to understand better why that is. And so I knocked this gene out of several pancreas cancer cell lines that expressed it, and found that those cells that lacked expression of Lin28B had a much less aggressive phenotype in terms of their ability to form colonies in soft agar, in terms of their abilities to invade and migrate in cell culture systems. In addition, I found that these cells, when implanted into mouse models of cancer, were less aggressive, less metastatic, and less proliferative. Part of the reason why we think that this is occurring is the overactive activation, let's say, of a maladaptive stress response, which causes characteristic gene expression changes and increased sensitivity to chemotherapy in culture models. And the other finding that we found from the sequencing was that the expression of this protein seemed to correlate with many kind of bad actor gene sets that were already published in the pancreas cancer, including the collagen quasi-mesenchymal gene set, which correlates with very poor outcome. And so basically what we found was that the presence of Lin28B sequesters and inactivates LET7 microRNAs, which then doesn't allow those LET7 microRNAs to be protective, and that conversely, the absence of Lin28B de-represses LET7, and that lets that state of the cell be less aggressive. And so we're hoping that this pathway will become a new biomarker in this disease and perhaps other solid tumors, and that perhaps we can develop small molecule inhibitors of this pathway to modulate its function and to have a new drug option for this disease.